Welcome to Easy Microbiology.
Good morning, everyone. Today I'd like to present to you my findings on Actinomyces naeslundii, a facultative anaerobic bacterium. Through this presentation, I will discuss the characteristics, habitat, and pathogenesis of this bacterium and how it can contribute to the development of dental caries and periodontitis. Let's get started. Actinomyces naeslundii is a common gram-positive, facultative anaerobic bacterium found in the environment and belonging to the Actinomyces genus. It plays a significant role in breaking down various organic matter and influencing the neuron system. This slide provides a 3D rendering of the neuron system to better comprehend its function in the ecosystem. A common commensal species of the human oral cavity, Actinomyces naeslundii, is often found in dental plaque and is related to the formation of both dental caries and periodontitis. As such, understanding and recognizing this species can be important when discussing and emphasizing the importance of oral hygiene and regular dental checkups for reducing occurrences of these conditions. Actinomyces naeslundii is a non-motile, rod-shaped bacterium that forms long, branching filaments called hyphae. Visible to the naked eye, its colonies can grow to be quite large. Under a microscope, one can observe the long, string-like structures of the hyphae, with this species of bacteria being one of the few that form them. Actinomyces naeslundii can be observed as white, raised colonies on anaerobic blood agar plates, with its shape being often irregular due to lack of oxygen. Such species of bacteria can prove to be beneficial in determining the presence of other bacterial strains. A facultative anaerobe, Actinomyces naeslundii has a variety of biochemical characteristics. It is capable of fermenting carbohydrates like glucose, fructose, and maltose, as well as producing enzymes like proteases and phosphatases. This reflects the species' high versatility, making it a valued microorganism in biochemistry. A species of bacteria known as Actinomyces naeslundii is typically found in the oral cavity of humans, making it a crucial organism to consider when it comes to oral health. This bacteria has a very limited range and is rarely found outside of humans, thus any research or knowledge pertaining to it could have a significant impact on the study of oral health. Actinomyces naeslundii is a bacterium commonly found in biofilms associated with dental caries and periodontitis. Virulence factors produced by the bacterium can cause tissue degradation and aid in biofilm formation through polysaccharide production. This can result in significant damage to dental and gum tissue and is believed to contribute to the development of dental caries and periodontitis. Achieving resistance to various antibiotics is an important factor for Actinomyces naeslundii. The utilization of efflux pumps to remove the antibiotic from the cell and the modification of antibiotic target molecules allow it to become resistant. Additionally, certain genes can be mutated which contribute to an increased level of resistance. It is important to be aware of the risks posed by Actinomyces naeslundii, which is typically found in the oral cavity of healthy individuals but can also lead to the development of dental caries and periodontitis. To limit risk of infection, proper dental hygiene and regular visits to the dentist should be undertaken to help maintain oral health. A. Naeslundii is a common species of gram-positive, acid-fast bacteria found in the human mouth. It is known to contribute to oral health problems such as dental caries and periodontitis, as it colonizes dental plaque and produces a range of virulence factors that can degrade tissues and promote the formation of biofilms. Consequently, A. Naeslundii can be a major factor in dental plaque-related diseases. Immune system of the body is essential in protecting against Actinomyces naeslundii. Antibodies are generated to combat the infection and many immune cells are activated to target and eliminate the bacterial infection. This results in a better defense against this bacterium. Actinomyces naeslundii is a species of bacteria that can be identified through microbial culture as well as molecular techniques such as polymerase chain reaction, PCR. Its importance in the medical field lies in its ability to cause infection in areas such as the mouth and genitals. Accurate diagnosis of Actinomyces naeslundii enables effective combat of these infections and provision of necessary treatment. Maintaining regular brushing and flossing, combined with a reduction in intake of sugary foods and drinks, is critical in the prevention of Actinomyces naeslundii associated dental caries and periodontitis. By maintaining good oral hygiene and limiting sugar intake, you can reduce your risk of developing these diseases. Removal of the affected area through scaling and root planing is necessary to treat Actinomyces naeslundii, a type of bacteria linked to dental caries and periodontitis. Antibiotics and antimicrobial agents are then used to target the bacterial infection, helping to reduce the severity of symptoms and control the infection. Conclusion drawn from this presentation is that Actinomyces naeslundii is a significant organism living in the human oral cavity. In the event of existing in an excessive amount or not following basic oral hygiene, it can lead to dental caries and periodontitis. Prevention and treatment of such conditions can be achieved by way of good oral hygiene, targeted biofilm removal, and antibiotics application. Let us remember to look after our oral cavity, keeping it clean and healthy. Thank you.